Hello, everyone, and welcome to today's live broadcast, a keynote presentation, Supporting Precision Medicine and Clinical Research at Scale, Challenges and Opportunities. It is presented by Niall Lennon, PhD, the Senior Director of Translational Genomics and Product Development, and an Institute Scientist at the Broad Institute of MIT and Harvard. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Lennon. I will now turn the presentation over to him.
Thank you, Judy. So it's a pleasure to be here to give this presentation to you today. The premise of my title and my presentation is that precision medicine is at an inflection point, moving from a concept at academic medical centers to a reality across a broad swath of the healthcare environment, and towards being an accepted standard of care in several areas. But what is the evidence for that? Is this true? Are we really at a turning point? There's certainly a lot of news about it. So I want to dig in a little bit on, how do we believe that this is true? And I guess to think about that, we really should think about how are we defining precision medicine today? What does it mean? To me, I first really sort of thought deeply about precision medicine in the oncology space, as many people have. And this review by Levi Garraway, formerly of the Dana-Farber and the Center for Cancer Precision Medicine there, in 2013 sort of summed it up nicely for me about precision oncology, and really shows the thinking at the time, which was that molecular profiling of a patient's tumor could lead to understanding of individual drug regimens that patient might particularly respond to, or might inform clinical trials that patient could be enrolled in, or could be prognostic of a patient's outcome. And so this was really, for me, sort of started to think about this personalized precision. We moved from the definition of personalized medicine to precision medicine, using genomic molecular profiling to help guide therapeutic choices or patient enrollment. In the 5 years or so that have followed that, we've seen tremendous activity in this space, and several companies have now launched and received FDA approval for genomic profiling tests. Some more notable ones are the Foundation Medicine test and the Oncomine test from Thermo Fisher. So these tests have received FDA approval now as companion diagnostics, which allows them, this genomic profiling, to be used to help assign people to particular treatments. And more recently, we're actually seeing CMS, Center for Medicare and Medicaid Services, issue some coverage guidelines for this type of testing. So this really, as we start to see reimbursement for this type of testing, this really moves it from the self-payer, limited access model to really more broad, widespread adoption. So that's certainly a good indicator for the field and adoption. Another indicator is that we see healthcare systems start to launch projects and programs to make this type of testing more broadly available. The VA, Veterans Administration, is one such program that's recently announced. It is expanding some pilots that they've done in precision oncology to to make this type of testing available to all of their clinical sites. And this is as one of the largest healthcare systems in the country, this is a large move towards the adoption of this type of testing. Just last week, however, there has been some notes of caution. We have to be very careful, according to this editorial in Nature last week, about overhyping the types of personalized treatments that can be assigned based on precision medicine testing. We're all aware of the great cases of immune checkpoint inhibitors literally melting away patients' tumors. However, there is much research still to be done about what patients will respond to such treatments, what patients become resistant to such treatments, and the mechanisms underlying both of those scenarios. And so overselling the promise of precision medicine too soon could also be very detrimental to the field. So that's a challenge that we need to be cognizant of. Of course, we all sort of thought of precision medicine mostly in the cancer space in the last couple of years. However, this is now really moving into the more common disease, non-cancer space as well. One program that's really challenges us to think about precision medicine at scale is the All of Us research program. This is NIH's program, formerly known as the Precision Medicine Initiative. And so really this seeks to enroll up to 1 million participants across the United States in the next 5 years and do genomic profiling of everybody. And from this 1 million person cohort, there will be many, many, many research things that can be done, including understanding the relationship between genetic risk factors and certain diseases, being able to monitor, because there's association with clinical records, monitor response to different types of treatments, enroll people in clinical studies for different disease types. So this is a very valuable cohort going forward. And there is an element of return of results to each individual participant here, so that each person can use the genomic information that's being generated in some way. So this is, again, a definition of personalized or precision medicine in action at scale. I should say, on a personal note, I've enrolled in the All of Us research program. It is open to everybody, all of us. And so blood collection, sample collection, clinical checkup, very simple. I did it locally at the Mass General, and I'd recommend anybody who's interested in getting genomic data about themselves enroll in the All of Us research program. Of course, there have been some commercial companies launched in the last couple of years who are really providing access to precision genomic information to individuals. Probably some of the more famous ones are listed here. 23andMe, of course, folks know, direct-to-consumer genetic testing, allowing people to understand many attributes about themselves, including not just ancestry, but also risks for developing certain diseases. Companies like Invitae and Color Genomics are providing very focused testing for particular risk factors in certain diseases. And recent studies have shown that this direct-to-consumer genetic testing is likely to continue to grow over the next couple of years. And again, this is scalable genetic testing with the purpose of providing personalized information to folks to make healthcare and lifestyle choices. So this is exciting. There are non-commercial activities in this space as well. One example of this is the Count Me In program that's run from the Broad Institute. And this actually seeks to find and enroll patients from particular disease type, who have a particular disease, into research studies, and to really change the paradigm about how enrollment on clinical research studies is done. Typically, historically, this would have been done at the local hospital level when a certain patient presents with a particular disease. A clinical coordinator, asks them if they want to enroll in a research study. And this has been the model across the country for many, many years. What the researchers realize is that if you're studying particular subsets of patients, those are maybe patients with metastatic breast cancer who have a particular response, exceptional response to some drugs, or on the other side, people who become resistant to their treatments. You really want to study these mechanisms, as I said before. But at one single hospital, you likely don't see enough patients, even at a large hospital, in these subsets to do rigorous, robust, scalable research. And so the Count Me In project allows participants to enroll from around the country. Metastatic breast cancer project was one of the first and most widely adopted of these types of projects. This project has now enrolled over 4,500 men and women with metastatic breast cancer from all 50 states in the United States. And these folks have logged on to the mbcproject.org website. Largely this information about this study is passed through social media, events for patient groups, and word of mouth. And so people log on. When they log on, they essentially fill out questionnaires. They give consent for the researchers to look at their medical records, and they give consent for the researchers to contact their primary doctors or their local hospital to receive, request some of their tumor biopsy material if it exists. And they also receive saliva tubes just from us, just as they would as if they had signed up with 23andMe or one of the other direct-to-patient genomic testing companies. And we use that saliva we collect as the sort of matched normal material to compare against that patient's tumor. And this helps us to do better, higher sensitivity mutation calling of their tumor. And so this molecular profiling then on every patient allows us to identify subsets of patients and do the underlying research. And so this is a really sort of great paradigm for enrolling large sets of patients to do really meaningful research. And so we see, again, going forward, we see a lot more involvement of individual participants. They're no longer just anonymous, faceless patient cohorts. Now these are really, each patient is invested in their own disease and their research, and they feel like in some small ways they can contribute, and they are making a large contribution to the research that may help, even if it doesn't help them, can help future generations of folks with these diseases. And so this is a very worthwhile sort of paradigm shifting approach to sort of personalized involvement in research. Of course, once we get into this precision, personalized world of genetic testing for everybody, we can learn all kinds of things about ourselves that maybe don't fall into the precision medicine bucket that we originally thought we would be in. There are several groups now who allow sort of direct to consumer genomic testing for lifestyle information. And Vinome is one example here of a group who will tell you about your wine preferences based on your genetic makeup. This is precision something, and totally valid. But also as we expand testing, make it cheaper, make it broadly available to everybody, there's all kinds of things you can learn, not just relating to our health. Another metric we may use is who are the big winners on the technology side. And so Illumina, many of us are familiar with as one of the major technology providers for genomic and genetic testing, both on the genotyping array platforms, which a lot of the 23andMe and others use, and the next generation or massively parallel sequencing field, which is used in larger genomic studies. And so, a reflection on the growth of that company over the last couple of years can really help us look at, wow, something is changing. The community is certainly growing. The market is certainly growing. Okay, maybe a non-scientific piece of evidence about the state of this field. I am really, really busy. Why is that a good marker of anything? Well, that really has to do with what I do and where I sit. So some people may be familiar with the Broad Institute. If you're not, it's a large nonprofit institution in Kendall Square, Cambridge, just outside Boston. And our mission is to transform medicine using systematic approaches in the biological sciences to accelerate the understanding and treatment of disease. The way the Broad is structured is that we have research programs that are focused on particular disease areas, including cancer, infectious disease, population and medical genetics, psychiatric disease, and others. And then we have large-scale technology platforms who build infrastructure and processes at scale to serve all of the various disease areas. And so it is within the Genomics Platform at Broad that I sit. The Genomics Group at Broad encompasses the largest single group, probably over 200 individuals now. And it spans everything from wet lab, sample saliva, blood, sample receipt through sample preparation, data generation on sequencing platforms or array genotyping platforms, primary analysis, base calling, secondary analysis, variant calling from this data, and then various ways to present and share data across researchers, host data in a scalable way, and we'll talk a little bit about that later. And within the Genomics Group, then, the Genomics Platform is the data generation powerhouse here, which encompasses over 100 folks and sits on one of the larger install bases of massively parallel sequencing platforms in the country, with largely an Illumina-centric fleet. But this actually allows us to undertake very large projects. And so Broad is probably known among those who know it for the scale of the projects that are undertaken here, dating back all the way to the precursor projects from the founding of the institution at the Human Genome Project, through large-scale data generation projects for 1,000 genomes, the Cancer Genome Atlas, TCGA, the Human Microbiome Project, all the way through things that are just starting up now, the Human Cell Atlas for single cell sequencing across many different cell types. And so this has really driven data generation across a number of different assay types here. As you can see in the graphs here, we've really sort of seen exponential growth in the last couple of years in the amount of data we've been producing, largely driven by the increase in the number of whole human genomes that we've been sequencing. We just recently sequenced our 100,000th human genome. Let's take a moment to absorb that. The first human genome project that was done here in collaboration with many groups around the world took around 13 to 15 years to get a draft done, cost about $3 billion. Now we're producing hundreds of human genomes every single day for a cost around $1,000. So that's been really part of this genomics revolution that underlies the application, the translation now of this field into healthcare and medicine. We've also done over 1 million of the genotyping arrays that allow for risk status, genome-wide association studies, exome sequencing. There's a method that has been developed at the Broad, has been applied here over 350,000 times. And we also do large amounts of RNA sequencing, gene panel sequencing, and other things. What's maybe less well known about the Broad is that we actually also have a clinical sequencing arm. We've a couple of years ago created the Clinical Research Sequencing Platform, or CRISP. And this platform is a CLIA CAP compliant lab that allows us to do clinical sequencing. It is always designed to be relatively small scale to start and really serving the clinical research community that we interact with here with our local hospital groups and across the country. And so we've done around 15,000 clinical samples in our clinical lab here that across some small number of tests that include germline and somatic whole exome sequencing. We have one non-sequencing based test actually for a rare kidney disease. And we've done many custom gene panels for cancer and germline sequencing as well. And as of March 1st, we've actually just launched a CLIA whole human genome sequencing test as well for germline indications. Again, largely, it's not really our mission to be a send out diagnostic testing lab. We're really here to support translational studies. And so we've done that over the last few years, supporting over 14 different clinical trials, ranging from small investigator trials at local hospitals. One that is very much along the lines of precision medicine is a trial around the development of personalized cancer vaccines for patients. And that's recently been published, as well as some publicly released information around major pharmaceutical companies that we've worked with to help with their clinical trials as well. So what are people talking to us about right now? Why am I so busy? Well, people are talking to us about developing lots of different things in lots of different areas. But I thought maybe two particular case studies could be useful to illustrate where we're at in the field and where we could be going. And so these case studies really are one on the cancer side. We'll talk a little bit about liquid biopsy, and one on the germline side, clinical human genome sequencing. So many folks are probably familiar with liquid biopsy for cancer at this point. To summarize, traditional biopsies have really been around surgical resection or fine needle aspiration of tumor material. And from that, the traditional paradigm in pathology has been to do, formalin fixation, paraffin embedding for immunohistological examination. And so a lot of the archived and even new tumor material we get are in the form of FFPE slides. These can be challenging, of course, as we know, for genomic testing. There are also the ability to collect fresh frozen tissue as well, and that can be much easier to work with from a genomics perspective. However, in the last couple of years, we've seen a large groundswell in interest in collecting liquid biopsy. So this is, traditionally today we're talking about peripheral blood collection, although there is work on urine collection as well. And so obviously the benefits of being able to work from peripheral blood is that it is cheaper and far less invasive to the patient. And it is possible to continue to collect peripheral blood even after a patient may have had a tumor removal with curative intent. And this maybe allows us to monitor patients post-surgery for any residual cancer or metastatic cancer. So the challenges, of course, over the last couple of years have been, how do we, from peripheral blood, examine or genomically characterize a patient's tumor? And so there's been a ton of work in the field to show that we can extract from blood analytes that actually reflect the tumor. And these analytes have been in the form of circulating tumor cells, which maybe 3 or 4 years ago had a lot of attention. And then more recently, that's shifted actually to cell-free circulating tumor DNA. And the reason that the shift has happened to maybe cell-free has been around, again, the ease of collection. It's a specific extraction protocol as opposed to circulating tumor cells, where you need more specialized equipment to capture those cells. And then you're dealing with a very small number of cells that you need to do specialized molecular biology on. So a lot of focus has been on cell-free DNA in recent years. There's been a large activity at the Broad Research Group in our cancer program that's been looking at that and has developed some strategies for looking at blood-derived cfDNA. And there's a paradigm that we're working on now, which is collection and extraction of cfDNA from whole blood, and then application of ultra-low pass whole genome sequencing. So this is genome sequencing at the less than 1x coverage of the entire genome. From that, we've developed analytical methods to be able to estimate the amount of circulating tumor cell in that cfDNA sample. Based on the amount of material there, the sample then may be suitable for differing downstream genomic assays. If you have a large amount of circulating tumor, greater than 10%, you could do whole exome sequencing or whole genome sequencing. If you're in the 1 to 10% range, then it's more suitable for a deeper gene panel type sequencing assay. And then if you have very rare, very low levels, you may need an additional sort of very personalized type of assay. And this is just information from a publication recently from Victor Adelsteinson, who's led that cancer program research group at Broad. And on the left, we're seeing in the top 2 panels here the close correlation between ultra low pass or 0.1x whole genome sequencing and a more traditional deeper genome sequencing from the same patient. And what we're looking at here is a copy number profile across the entire genome. And what you can see is that the copy number spikes, the amplifications and deletions mirror exactly between the ultra-low pass and the deeper genome sequencing. And from these, we can estimate tumor fraction. And you can see the tumor fraction estimation is exactly the same in this case, in the same patient. And the bottom two panels, we're also seeing that even at 1x of that same patient, from the tumor now, not from the circulating free DNA, we're seeing, obviously, we've got a higher tumor fraction because now we're biopsying the tumor directly. And we see correlation between genome sequencing and whole exome sequencing of that tumor in the copy number profile. And all of this is from one patient. And so you can see across the cfDNA and the solid tumor, we see the same pattern of copy number. And in the very low pass whole genome, we're able to see this pattern, and we're able to estimate tumor fraction in that cfDNA. So this is a very powerful technique that allows us to triage incoming blood samples for to understand what type of downstream assay is most suitable, and also what samples are likely to succeed and what samples will not. And this is important as an acceptance criteria if we move this to a clinical testing paradigm. On the right-hand side here, we're seeing then looking at matched tumor and cfDNA genetic alterations in a group of patients, and we see really good, on average, correlation between the variants found in the tumor and in the circulating free DNA. This is important as well if we want to make inferences about a patient's tumor from whole blood. To get technical for a moment, we've over the last year in our R&D group spent a long time understanding how to better access these very low amounts of circulating free DNA and the even smaller amounts of ctDNA we get. We haven't talked about it yet, but from whole blood or plasma, we may often only get 5 to 20 nanograms of material out. This is a very small amount to start any kind of genetic testing from, or genomic sample prep. And so, and within that 5 or 20 nanograms, only a subset of that will be the tumor DNA. And so you really need methods that allow you to have, one, a very high conversion efficiency of input molecules to final sequenceable molecules. And also we need to, because we have to amplify these molecules to sequence them, we need to incorporate methods that allow us to distinguish duplicate molecules from unique molecules, and to be able to do variant calling with high sensitivity at low allele fraction. And so there's a lot of requirements on both the molecular biology and the analytical side that need to happen to make that really work well. And one of the tricks that we've seen that works the best is actually switching to this dual Y adapter with dual indexes and dual UMIs, unique molecular indexes. And that's very important for downstream calling. We've, obviously, ideally, you'd be able to do development on many patient samples, but because of the scarcity and valuable nature of real patient samples, we've created synthetic models of low allele fraction events by taking healthy donors. So healthy individuals actually have circulating free DNA. It is not tumor DNA, but it's cell-free DNA. And so if we take two unrelated donors and spike one person's cfDNA into the other, we can artificially create a model that looks like a ctDNA at varying levels. And then we can characterize many variants within that set, depending on the territory that we sequence, and from that calculate our sensitivity and our specificity. And so there's a lot of work ongoing here, but one result we can share here that is illustrative of the types of things that we have to optimize for. Using traditional library adapter systems, we were seeing very high false positive rates. And that's illustrated in these Lego plots. If you haven't seen a Lego plot, this essentially just shows all of the different variant types within a given sample and the base context in which that variant type is seen. This plot is very useful if you're looking for a particular type of artifact in your sequencing, things that have been seen, say, FFPE samples. But illustrative here is that the height of the bars in this Lego plot are telling us that there's a lot of false positives in our data when we use traditional library construction methods. When we switch to our latest duplex dual UMI adapter methods that have been published by many groups now, we see that this actually, coupled with our analysis methods, drastically reduces the number of false positives. And this is very, very important when you're doing genetic testing, especially in the clinic, because we really want to be able to extract real meaningful signal from false positives that will send you down the wrong path. So that's just a really quick overview of some of the things we've been doing in blood biopsy. And many groups are doing this same type of work. And so some of the challenges here and considerations for widespread adoption of this method are understanding at the pre-analytical steps. If we want to do liquid biopsy-based testing on patients, we have to start to think about the process at the collection point. So how are we collecting the sample? How much sample are we collecting? Is it possible to collect 2 tubes of blood instead of 1 and give us more molecules going in, which gives us higher complexity libraries, which gives us better sensitivity? The types of tubes we're collecting the blood in is important. If we're collecting in traditional types of EDTA tubes, then the blood has to be fractionated rapidly to avoid degradation of the cfDNA. There are specialized tubes that you can use to collect blood that makes it more suitable for downstream cfDNA extraction, and so that needs to be adopted. We also need to think about what I talked about, the sample prep and the analytical methods and how we scale those across many different gene panels that people want to use now in different tumor types. And so this is something that we are thinking about actively here. And then, of course, we have the sort of the more at the higher level, understanding the real clinical utility of this type of testing. Many groups have shown this high concordance between cfDNA and tumor biopsy, but what level of concordance is required to really be clinically useful, and what can we do from these studies? And so clinical utility studies are really where we're at right now and across a lot of different groups to understand the different utilities, whether it's diagnostic, prognostic, or in monitoring minimal residual disease in patients post-surgery. And so very active area across in the cancer space. So switching gears, thinking about more on the common disease side, the germline Mendelian disease side, thinking about whole human genome sequencing. It's really only been in the last few years that we could contemplate human genome sequencing, whole human genome sequencing at scale. Previously, we've been largely doing panels or exomes, and that's largely been driven by cost. But as the technology platforms have advanced in terms of yields per cost of those yields, we've been able to drive down the cost of sequencing, and that really has increased adoption initially in the research space to generate data for us to work from, but now increasingly into the clinical space. I think there may be a perception by some folks who aren't in the trenches that now, whole genome sequencing is just a solved problem. Anybody can do it. You just buy a box from Illumina and you're good to go. And I really like this quote from Ewan Birney, who's the director of the European Bioinformatics Institute, on Twitter a few months ago about genome sequencing and comparing it to say it's routine in the same way that the US Navy routinely lands planes on aircraft carriers. Yes, a good organized crew does it routinely, but it is complex and surprisingly easy to screw up. And having done 100,000 of these genomes here, we can tell you that it is still a complex process and can go wrong in many, many ways. And we've seen a lot of those ways here. And so from that experience in research, we've been able to take that process and make it suitable for clinical use. To do that, we've taken our research pipeline and gone through the process of making it CLIA compatible. And for folks who've gone through that, this is really about addressing your process, your people, your equipment, every part of the end-to-end infrastructure, establishing analytical and technical validity of the method, documenting it, and making sure that you can do it robustly, reproducibly. And so we've gone through that. And like I said, in March, we launched our whole genome. We took our research whole genome process and we made it from March 1. It could be used for CLIA purposes also. Of course, as you're doing that, you have to make some decisions. And one of the things that people who are doing clinical whole genome sequencing still discuss is, well, how much sequencing should you do? What depth of coverage of the genome is meaningful? And this is a continuing debate, and how we measure coverage is not necessarily standardized. This is one of the challenges across the community. And then what is good enough coverage is another debate in the community that needs to be sort of standardized or harmonized for widespread adoption. What you see here is really just a quick snapshot of understanding the relationship between, on the right-hand panel here, mean coverage of the genome. So a lot of folks think about, a default human genome being a 30x human genome. That's been a number that's been used in research for a long time. Other groups would say to be clinical, you need to be more of a 40x genome or a 45x genome. A couple of points there. One point being that just mean coverage as a single metric is probably not the right metric for us all to use in the community, because just looking at one number like coverage can be misleading. You can have 30 or 35x coverage of a genome, mean coverage, but have very uneven coverage within the genome. So you really need to think about evenness of the genome. And one way to account for that is actually thinking about the percentage or proportion of your genome that's covered to a particular target coverage. And so one metric we've looked at here is, what is the percentage of our genome that's at least covered at 20x coverage? We know from variant calling statistics that at 20x coverage at a given base, you are well powered to call at least a single nucleotide variant at that base. And so percent at 20x encompasses coverage not just across the average coverage across the genome, but actual situational coverage or evenness of that coverage across the entire genome. And what we see is that, in our clinical validation study, our target has really been a minimum of 95% of the genome covered at 20x. And what we on average see is that we're really getting closer to 97.5 or 98% of the genome covered at least 20x. And that correlates to a mean coverage somewhere in the 35 to 40 range. And so that's typically what our genome looks like. And here we can see the relationship between those coverages and sensitivity. The blue line is for SNV sensitivity, and the orange line for indel sensitivity. And of course, you can see that going to higher coverage gives you higher yields or returns on sensitivity for particular variant types. But then you also have to balance cost. So cost is a huge factor in applying any kind of genetic testing at scale. And so we're in this area of balancing performance and cost. And again, this is somewhere where we need to decide on the right targets across the community. One way to think about that is, taking a subset of clinically meaningful genes and saying, well, how did these genes perform at your target coverages? ACMG 59 has been something that has been thrown around a lot and is also being used in the All of Us program. ACMG 59 refers to a set of 59 genes defined by the American College of Medical Genetics as genes that have important findings for a patient. So oftentimes these are known as incidental findings or secondary findings or returnable findings, but these are genes like BRCA1 or genes associated with cardiomyopathy, heart disease, particular diseases that if you find pathogenic variants in these genes in the course of your genetic testing, the ACMG recommends that you inform the patient about this. And there's a lot of debate at individual clinics and healthcare centers about what to return, what to consent people for, but these particular variants are important. And so looking at how do we perform on these genes, not just at coverage, but we then looked at callability as a different metric. And this is a metric or a modification version of a metric that's been proposed by you and Ashley at Stanford, to look at an individual base and say that the base is callable, that is, you could call a variant at this site, in a more stringent way of saying if it's covered at least 20x coverage, it has a mapping quality of Q20. That means you are 99.9% sure that read maps to that part of the genome. There's many parts of the genome that look very similar, and so sequencing reads could be placed at multiple sites, and so you want to be confident that your read is at the right site. And the base quality, that is, you are confident, again, 99.9% confident or greater that the individual base that you called at that site is correct. And so applying all three of these criteria, we can come up with a callability metric. And looking within the ACMG 59, we looked at all of the bases in those genes and said, how are we doing for callability on these genes? And if we saw, greater than 20% of our cohort not having a callable base at a given site, we say that base is not callable, even though many of them could be called, but this is quite a stringent criteria. And so looking at ACMG 59 again, there's approximately 16,000 or so pathogenic or likely pathogenic variants in this gene set, according to one of the more well-known databases of pathogenic variants, that's ClinVar. And we looked at those, all of those variants, and found that of the 16,000, only 20 or so were deemed not callable by our stringent criteria, and therefore in this genome that's between 30 and 40x, we're covering over 99.9% of the pathogenic variants in this set. So that's one measure of quality. Again, there's a lot of debate that in the community, and we need to come up with standards to decide what we believe should be the right metric and what the target should be. So, one thing to think about as well is there are several challenges now and considerations for widespread clinical adoption of whole genome sequencing. Like I said, even up until recently, it was cost prohibitive. Now we're seeing a trajectory towards it not being cost prohibitive when we compare it to exome sequencing or gene panel testing. But we really, to realize those costs, we need to operate at scale. And so we need to ensure this clinical grade quality at scale to serve the entire patient population. And so we've lots of case studies now, or many case studies now in the community of individual patients who've had maybe long diagnostic odysseys of individual gene testing where whole genome sequencing has found the answer. And there's lots of reports of the diagnostic yield of genome sequencing versus exome sequencing versus individual gene testing. There's also been, demonstrations of we can do genomes very fast, 24 hours from blood draw to data back, and in some contexts that could be important. But really, to be transformative, we need to do this at high quality and at scale and at a cost that a healthcare system can afford or can bear. And so, one-offs or demonstrations of speed can be done, but can be quite expensive. And so we need to think about how do we scale this? How do we do it reproducibly and robustly? And that's something that we've obviously thought about here a lot. Then, as you do this at scale, you're really talking about generating a lot of data. Whole genome sequencing generates a lot of data. Traditionally, individual sequencing sites would have stored that data locally or on-prem. That just becomes not feasible when you start to think about whole genome sequencing. Just the cost of storage and compute, it becomes quickly unmanageable for almost all institutions. And so we really need to understand where we host the data, how we do compute on this data in a cost-efficient way, and also getting away from the traditional paradigm of where, if you did a research study and you wanted to collaborate on some analyses, your collaborators would just download or copy your data and do their analysis. As we get to these very, very large data sets, moving the data around, copying the data becomes sort of non-trivial and really is sort of an inefficient way to do it. And so we, and others have been looking at ways to create environments where you can store the data and bring the researchers to the data or bring the clinicians to the data, as opposed to distributing the data. We also need to think about in the future, how much data do we need to keep? Is the raw data important? If we start this as a clinical test, what do we define as the data that we must keep for several years? Is it the raw data? Is it the variants that we've determined from that data? Is it the report that's been created from those variants? Raw data, we used to keep in earlier versions of the Illumina platforms, we used to keep image files. That's become untenable. We don't keep image files anymore. Now we keep the base calls. How long is it before we decide that maybe even keeping the base calls is no longer critical? A big open debate in the community, and something that we need to all sort of think about and think about methods for addressing. So at the Broad, of course, we've built a cloud platform for this. We've also, we're also known for some of the software tools, the genome analysis toolkit that we've made available on that cloud environment. The cloud environment is called FireCloud, and we've done that on both to serve both the cancer community and now the genome community. Data we sequence here, both for research and clinical, is made available through FireCloud, and we think that's where our future sort of clinical data delivery will be. Okay, so we've covered a lot of different things here on the cancer side, the germline side. I think clearly there's massive opportunity for the community at large to use genomic testing and have it improve patient outcomes in the cancer space by indicating more therapies or treatments that are more likely to succeed, or to allow better, more rational design of clinical trials to get newer treatments out faster. And on the germline side, it's an opportunity to really, at scale, understand risks for diseases and underlying mechanisms of the diseases. When we do very large numbers for common diseases, we need to look at very, very large numbers of patients, and we need to look at patients from across all types of different ethnic backgrounds, because inferring pathogenicity or frequency of particular variants when looking at just one ethnic background is not helpful. And so one of the things we hope the All of Us research program will do is actually increase our knowledge base of frequency of common and rare variants in across different types of backgrounds. And so that will also give us power to address common diseases going forward as well. Okay, that's a lot. So now maybe you understand a little bit of why I'm so busy, because I think about all of this all day long. So finally, I just want to thank you all for listening to this. Hopefully you found it interesting. If you have questions about sequencing projects or anything you would like to do with the Broad Institute, you can email us at genomics@broadinstitute.org. And there are, of course, this endeavor is really built on a foundation of many hundreds of folks who work here, who provide this high quality data and analysis. And so a thank you is due to all of them as well. Thank you.
Okay. Thank you, Dr. Lennon, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, can you discuss how a group might decide between sequencing a gene panel, an exome, or a genome for both cancer or non-cancer studies?
Yes, so this is something that comes up a lot. And, ideally we would say it's all about what gives you the scientifically best answer, but in many cases it also is driven by cost. And so on the non-cancer side, really cost has driven us to do exome sequencing and gene panel sequencing in the last 5-10 years. But what we're seeing now is that, as I described, with the technological advancements, we're getting whole genome sequencing down to a place where it could start to cross over, certainly initially with exome sequencing, and maybe at some point with panel sequencing, to be cost competitive. And we know that the data quality for germline purposes of a PCR-free whole genome is just far superior to anything that you'll do that really that has a selection step or an amplification step, because of the inherent biases that are introduced when you do selection or PCR. On the cancer side, it's a little bit different. For cancer, because of the stromal contamination of non-cancer cells in any kind of tumor biopsy, or the circulating free DNA that's not ctDNA in a liquid biopsy, the proportion of your molecules that come from the tumor are always lower. And so you need to be able to have power to call variants in that tumor DNA. So you need to go deeper. And so because you need to go really deep, that's really pushed the cancer space to panel sequencing to allow us to do far deeper sequencing on a smaller territory to be, again, cost efficient. But we are starting to see some genome sequencing in the cancer space. And again, what genome sequencing gives you in this context is the ability to address different types of structural variation that are really hard to address from panels. things like structural and large events in the genome. So right now in germline, I see a shorter term shift in some settings to genomes. In the cancer world, I see panels being sort of the way to go for at least for the short to midterm.
Thank you for that. What about interpretation and return of genetic results, and what will be needed for this to scale?
Right. Yeah, this is a huge thing, and we didn't have time to get into it today. I think, again, when we work with academic medical centers, we sort of take it for granted that we have access to genetic counselors, and we have access to genetic fellows who can do interpretation and molecular pathologists who can interpret this type of data. But really, for widespread adoption, what we need is really large-scale training programs to train cohorts of genetic counselors who can return these kinds of results and explain these complicated results to folks, and training of physicians and healthcare providers to also be able to understand them. And there are many platforms and companies now who are working on creating reports that are more understandable. But also we need training and harmonization across the community on the meaning of some variants. Even in the germline community, different groups who interpret a particular variant might interpret it differently. Some might interpret it as a benign variant, doesn't do anything. Some may say it's likely pathogenic. It actually might be causing the disease. And so harmonization of those activities across different clinical sites is very important as well.
What is the biggest challenge facing precision medicine today?
Right. So there are lots of challenges. Obviously, a lot of what I think about are the technical challenges, the implementation challenges, how we develop the methods to give us the best possible data, how we deliver and manage that data. But across healthcare systems, of course, it's really an adoption question about cost. How do we get the cost so we're not just adding to the crazy cost of healthcare already? And to do that, how are we demonstrating utility? So how can we show that if you spend the money on genome sequencing, you're saving money over the course of a patient's life by not having to do multiple individual gene genetic tests later on? So that clinical utility and cost-benefit analysis is ongoing in many groups, and that's really, I think, one major thing that has to happen. And the other is, on that technical side, how do we scale the testing, and then how do we integrate the results of the testing into medical records and into a way that can be absorbed and understood by healthcare providers.
Thank you. We have time for one more question. How long before we are sequencing everybody at birth?
Yeah, that's interesting future prospect, right? And so I'm conflicted on this myself. We've been part of studies that one study out of Harvard and Brigham Hospital called BabySeq. And this actually does do, in a small cohort, sequencing of babies from healthy babies and babies from the NICU, sick babies. And the study itself is really to address this exact issue. Well, one of these issues around what is the utility of that? What do you learn? Could you do something for the sick babies? What is the benefit to the healthy babies? And what is the process of actually consenting people for that? And so that's an ongoing study. And very interesting. And I think, from a purely scientific side, you could see a benefit of sequencing very early to identify risk factors for diseases that might be childhood, early onset diseases that you would want to know about, or later onset diseases that could be, risks could be ameliorated through lifestyle choices or more frequent screening. That could certainly be a benefit. I think to get to that point, we really have to address some significant concerns about privacy, genetic privacy, security of your data. How do you keep that, very, very personal data secure, and policy. Right now we do have a genetic non-discrimination act that says insurers can't refuse coverage based on genetic profiling information, but laws can be changed. And so what is the protection in the future for somebody who has that type of testing done? So these are big sort of ethical, legal, and social issues that again, many groups are looking at, but will be very interesting to see how it plays out over the next few years.
Well, I would like to once again thank Dr. Lennon for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September 21st, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.